so now let's turn our attention to polymerase chain reaction. So if you looked at the video on gene cloning with the vector, then you would know that PCR is another way to make more copies of genes. But the difference here is you don't need a vector, okay? So what you use is a primer and then a polymerase that allows you to make multiple copies of a gene. How do you do it? So you do have to know your gene. You have to look up your genomic sequence, and you have to identify sequences that come before and after your genes. So sequences that flank your genes, and then you're going to create primers that are complementary to those areas. And think back to what a primer does during DNA replication. So PCR really is like, it has found a way to take natural DNA replication and do it in a machine. So you have your primers, and they go to the areas that they're complementary to, and then DNA polymerase comes along and elongates from those primers. And so the process of PCR actually only has 3 steps. So you have your primers. So first thing you do is denature your DNA. You turn the temperature up really high to cause the double strands to come apart. This is what helicase does in DNA replication. So now you have these 2 single strands of your DNA molecule. And then you lower the temperature and allow for your primers to go and attach to their complementary regions. Now, after your primer is attached to the regions, then DNA polymerase can come along and elongate those primers, okay? And so that's your primer extension. And that's usually carried out by Tachman, and Tachman is a special polymerase because it doesn't denature when the DNA, so at high temperatures, Tachman doesn't denature. And if you think back to enzymes, they have very optimum regions, and most enzymes would denature, but this one does not. And so then you do that process over and over again. So denature, annealing, and extension are all one cycle. It happens one time, and then in PCR, you'll do it over and over again. What does that mean? After extension, then you up the temperature again, reduce the temperature, go up a little bit more. That's your second cycle. Put the temperature up again, and you're allowing all of these reactions to occur over and over and over again. Why? Because each time you're doubling the DNA, right? So when we first started, we had 1. When we end here, we have 2. Now then, each of these are going to be allowed to be doubled, and then this goes on and on, however many cycles it takes to get however much DNA you need to create. What makes this relatively easy or simple is that a thermocycler or a machine takes care of all of this for you. So what you have are these PCR tubes, and you just put everything in it. So you add the DNA, you add the primers, you add the Tachman, and then you add DNTPs, which are like free nucleotides that can be used. And then you put it in the cycler, you program it to tell it the temperatures you want it to reach, how many cycles you want it to go for, and then what to do when it turns off. And what I mean by that is some of them have a cooling feature so that you can keep this at cool temperatures when the process is over with to make sure the process does stop. Okay, so it makes it relatively simple. So after you get that, what then? So depending on what your end result is or what you want out of this, sometimes you run it on a gel. So this is gel electrophoresis. You have an agarose gel, you have lanes that you can add the DNA in, and then you run electric current to it, which will cause whatever you put in to separate according to size, and then you stain the DNA with ethidium bromide or some kind of a dye that allows you to distinguish one from another. So in either the first or the last lane is usually a ladder, and it shows you the size of your DNA, and then you load your different samples, and you're able to see what your product is. And sometimes this is used so you identify the product and then cut it out, purify it, and go sequence it, or whatever it is you're doing this experiment for. So that's gel electrophoresis. Now, like most techniques, there are alternatives. So one is reverse transcriptase, right? So we've talked about reverse transcriptase. So going from mRNA to cDNA. You can do this with PCR too. So here you'll have to add reverse transcriptase and a primer near the 3' because you know at the 5', a primer that added to the 3'. Now, this is your RNA of interest. You have to have that reverse transcriptase to turn it back into DNA, right? So this is different from the other one in that you have to make that cDNA from your mRNA, Okay, but this again allows you to look at expression of genes now. Okay, so PCR, you're going to amplify genes. If you look at reverse transcriptase PCR, you're amplifying gene products, so that mRNA that was made from your gene. And yet another process you can do is real-time PCR. Now this one is going to allow you to quantify your gene or your gene product in a mixture. So now here you have an oligonucleotide, which is like a primer, but now you're going to call it a probe. This TACMan probe has a reporter that fluoresces, and it has a quencher that absorbs that fluorescence. Okay, and this is what you're going to put in, and this is complementary to an area on your PCR product. So then what you do is you allow them to bind to each other, and then you have your primer. Well, when polymerase comes along and starts doing its job, it cuts this reporter from the quencher, which allows that reporter to then fluoresce. It's no longer being quenched, it's able to fluoresce. And that's important because the machine that this is carried out in is quantifying that fluorescence, right? So as you're cutting it off and it's fluorescing, it's emitting a signal that's being recorded by the machine to tell you how many times this is occurring, which is going to correlate to how much product is there. So this has phases. So that exponential phase where it's just getting started, remember we are doubling with every cycle, so it's just getting started. And then after you reach a certain threshold, when the fluorescence that's emitted by the product is more than what's the background, okay? It's just not noise. Now we actually have something going on. Then it's going to take that linear growth, and then it plateaus, okay? Now this is important because once it hits that exponential, you know, okay, we're looking at real product. This thing has gotten started. Now it's off to the races. So it's going. Well, we are only doing this as much product as we have. So here we're not amplifying. We just want to count what's there, right? So as we're counting it, and eventually each probe has bound to its complementary sequence, there's no more to do, right? So we start to plateau, like, all right, well, we're coming to an end because there's no more. And so that's how you count or quantify product here. You're going to allow all of your complementary sequences to bind to a TACMan probe and give off that signal. And so you're going through the different phases of exponential, linear, and plateau. You are comparing your sample, your unknown, to known concentrations. Okay, so you have some standards here, and you know what's in your standards. And so you're going to compare what's your unknown to your standards to figure out how much is going on here. Okay, so the more cycles this thing goes through, then you know that's the more product you're making, the more product that's there that you can attach to. So this CT number becomes very important. So that's the cycle threshold, and that's what's used when you're quantifying how much you have. And there are different formulas out there and different things depending on what your product is and what you're trying to get and what your standards are. That helps you to do this. But this is also an automated process. Now, I feel like this was more than the previous one. So remember with PCR, the first idea is you can use PCR to amplify genes. You can use reverse transcriptase PCR to amplify gene product. Or you can use real-time PCR to quantify mRNA or genes. Okay, so gene or gene product. So those are the different types of PCR or polymerase chain reactions. So one thing I tried to point out here is that PCR really is using the same steps of DNA replication. So we found a way to put that in a test tube, if you will. So what are some pros and cons to using this technique?